Hello, everyone, and welcome to ACS webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Michael David, and I'll be your host during today's webinar in celebration of National Chemistry Week. Today's webinar is being co-produced with CAS, a division of the American Chemical Society. Today, we have Chung Angela Zhao, the Manager of Scientific Analysis and Insights at CAS, as our moderator. Angela and her CAS colleagues have published multiple scientific trend reports on emerging topics such as COVID-19, lipid nanoparticle technologies, RNA therapeutics, and so on, all based on the CAS content collection.
Thank you so much, Michael. Hello, everyone. I'm very happy to be the moderator today for this exciting topic, the rise of targeted covalent inhibitors. Today, we are honored to have two speakers, Dr. Gary Gustafsson from CAS and Dr. Neil Devon from Totus Medicines. Before we get started, I'm just going to give you a very quick introduction of CAS, who we are, and what we do. CAS is an organization specialized in scientific information and solutions. We have hundreds of scientists working together to extract and analyze scientific data from a great amount of different sources around the world in more than 50 languages. As you can see, our content covers not only chemistry, but many other areas in biomedical sciences and drug discoveries. Every now and then, CS scientists also write and publish research work based on the landscape view of data from CS content collection. The landscape view of covalent inhibitors is one of the topics that our team recently worked on. So without further ado, I'm going to introduce my colleague, Gary here. And while Gary is bringing his slides over, and I'm going to introduce Gary a little bit of his previous research experience. Gary is a senior customer success specialist at CAS. Gary earned his PhD in organic chemistry from Brown University and performed a postdoc in biochemistry in the lab of Sidney Hatch at the University of Virginia. Gary spent the first 25 years of his professional career as a medicinal chemist in positions of increasing responsibility, and was a member of the project team that produced approved a drug called elucitinib, which is a drug for treatment of acute myeloid leukemia. While at Forma Therapeutics, Gary was also a professor of chemistry at Quinnipiac University and a Southern Connecticut State University prior joining CAS. Gary, thank you so much for coming, and the floor is yours.
Perfect. Thank you, Angela. And also thank you to the organizers of this webinar. Very excited to be able to share some research done by my colleagues and myself, whose names are on this slide, around the topic of covalent inhibitors. And I think you'll find by the end of this webinar that it is a fascinating and promising area that is increasing year over year. So to get into, let me start my slides now. All right. So we're talking about covalent inhibitors, and you're right, it's not a new topic. These have been around for a while. Reach way back to one that's probably known by just about everyone on this call, and that's aspirin, a non-steroidal anti-inflammatory drug. It acts by acylating the hydroxyl groups of serines and cyclooxygenases. And this then has the effect of decreasing inflammation through suppression of prostaglandin and thromboxane synthesis. Been around for quite a while, 125 years, and actually as recently as 2020, 17 million prescriptions were prescribed by doctors. However, this is not selective. So there are a number of off-target issues, most notably many stomach ulcers. If you already have asthma, that may get worse. And there are a lot of other ones indexed with this therapeutic as well. So that's an older example that's come a little more recent. Probably people are also fairly familiar with the clopidogrel, also called Plavix. And when dosed, it's actually a prodrug, and it becomes activated through its action on being active on bicytochrome P450, freeing up the file And then that does a thal bond with the cysteine and the ADP receptor. That keeps platelets from sticking together and forming clots. Before the patent expired in 2011, it was the number 2 selling drug in the world. They already talked about the fact that it is a prodrug. So this lessens the number of side effects, but there's still instances of thrombotic thrombocytopenia, occur, and also some instances of hemorrhage. So, and also to mention that these are both covalent alkylators or acylators, but it was not planned. It was something that was found out once it actually went into the clinic. So the issues that appear through many drugs that acted covalently kind of made a lot of companies shy away from going forward with something that is documented or proved to be a covalent inhibitor. But as time went on, what researchers found is that if they could attach a electrophile to some molecule that could get to the target where the effect was trying to be elicited, they could put it on the molecule just so that we put in just the right position to only form a bond once it was at the site of action. First example of this that I'm showing is a drug called afatinib. This was the first drug, and I haven't introduced the term of targeted covalent inhibitors, but you'll quickly see why, is that, this is being targeted to be only occurring in one place. So this was the first drug approved via this approach, and it acted through binding at the EGFR receptor and putting the alpha beta unsaturated ketone, which I'm circling right up here, in just the right location to react with the cysteine 797 thiol right here and forming that covalent bond. So let's talk a little bit about what the theory is behind these this class of inhibitors. So the molecule will basically have two portions. One portion will be the guidance system that will be targeted towards binding sites right near the locus of action. And then also attached will be a warhead. So upon binding at the site, that warhead is put in just the specific perfect orientation to react with a residue on the protein, which is being indicated by the star there. And then the covalent bond forms. So we've talked about one type of warhead, and that was the alpha beta unsaturated carbonyl. Literature will reveal that there are a wide array of warheads actually used. As they go across the top here, we see boronic acids, aldehydes. In the upper right, we have alkenes that are right next to an electron withdrawing group. Then across the bottom, we've got epoxides, alpha ketoamides, and then halogens, similarly also with a electron withdrawing group in the neighborhood. So, what advantages do we see or have been revealed in the literature about this class of compounds, especially when we're comparing it to non-covalent inhibitors? So, due to the very high biochemical efficiency and the fact that once it's alkylate, it's there, it's covalently bound to the protein, TCIs may require less compound that have fewer side effects. The covalent nature of it means that it's going to be there, and there may not be as frequent a dosing of this material is required. And oftentimes, depending on the site where the binding is taking place, it may be competing with an endogenous substrate. The fact that it's forming covalent bond gives it a very serious competitive advantage. Lastly, so the strength of the binding doesn't necessarily need to be as high because it's taking advantage of the forming that covalent bond. So now this opens up, and I think we'll hear about this in the next talk, to many undruggable enzymes that have shallow or non-discrete binding sites, specifically to mention a few, those involved in protein interactions. Now these can be targeted. So with that background, I'd like to actually go into the work that we did, and that's kind of do a lot of research within our content collection at CES to see if we can distill out any trends in the publications in this area. So and we're going to look at this information in another area, a number of areas, once again, mainly from the CS content collection. And we're going to look at such things as years of publication, what institutions were publishing the work, and then trends with regards to things like the warheads, the targets, the therapeutic areas, what have you. So the first thing we did was just kind of look at publications overall. And we've put this into two different bins, one being journals, one being patents. You can see the journals on the left, the patents on the right. It may not be a surprise to too many people here that journal publications were dominated by academic institutions, as you can see over there on the left-hand side. Correspondingly, if we looked at patents, these in turn were dominated by corporations, Merck, Janssen, AbbVie, or what have you. So that's the trends overall in target covalent inhibitors. Now, if we kind of map this out over time, we want to see if there are any general trends here. And the blue line will correspond to journal publications. The yellow line will correspond to patent publications. So key inflection points that we see here is going into 2011, 2012, there was a fairly substantial increase in the number of publications in the journal literature. And this may be due to the fact that the first compounds, the first targeted covalent inhibitors were entering the clinic. Probably a lot of publications in and around that. The second kind of interesting inflection point, it occurs up the 2019, 2020 timeframe. And what's interesting here is we actually saw journal publications go up, but patent publications go down. The explanation that we kind of got from several scientists that we talked to was a lot of scientists, at academic institutions were now operating remotely. They said this actually gave them a little bit more time to compile, their research together and put those journal publications out. With regards to the patents, I think everyone was adjusting to the reality of COVID, and may have taken institutions a little while to get the whole patent process together along with the legal firms. And that's why I might have seen a small downtick there, but it quickly rebounded, as you can see, in the next year. So this is looking at all publication targeted covalent inhibitors. I'm now going to put up a screen that just shows us the publications of the alpha beta unsaturated carbonyls. And if you think I didn't actually switch the slide, that's not the case. It's just that you'll see as we go on here, publications on the alpha beta unsaturated carbonyls will dominate or have dominated this field, at least over the first 10 years or so that they have been out there. So, and just to remind everybody what an alpha beta unsaturated carbonyl looks like, I just put this up here again, and you can see the functionality there in the circle. And as we already kind of discussed, it's the publication of molecules like this and their entrance into the clinic that probably put that first big inflection point up in the number of publications. So I'm now going to kind of parse out into a number of different graphs here, the other warheads that we've seen appear in the literature. And I've done this because, the alpha beta unsaturated carbonyls are dominating so much that you wouldn't really be able to tease anything out if I had them on the same screen. But I'm just going to kind of go through and show maybe what some of the other major ones were. Upper left-hand corner, you can see the aldehydes dominated that graph. In the upper right-hand corner, epoxides amongst the ones in there had the most publications. Lower left-hand corner, another technical alpha beta unsaturated carbonyl, these being alkynes rather than alkenes, also was one of the more prominent ones. And lastly, those that alkylated at a methylene that contained was bonded to a halogen, as well as a electron withdrawing group, had several publications as well, with fluorines being the most predominant. All right, so let's look at the data a different way now. And here we're just trying to tease out for the different warheads, which ones were responsible for most publications, that being the donut on the left, and which ones had the actual most number of registered substances. As we've already said, alpha beta unsaturated carbonyls are dominating these, responsible for almost half of the publications and actually over half of the number of registered substances in the target covalent inhibitors area. We then thought we'd take a look at and see what Lipinski characteristics this class of compounds had. And by and large, they're a fairly compliant group. You know, 82% of them either follow all 4 Lipinski rules or 3 out of the 4 Lipinski rules. And as you can see, that, or as we mentioned earlier on in this presentation, there's not as much a need for a super efficient binder there, because it is taking advantage of the covalent alkylation, bringing it to that spot. So maybe that combined with the warheads themselves are fairly simple, so that these may have led to lower molecular weight on average than maybe a lot of the other classes that are out there. We then wanted to take a look at some of the compounds that had either been approved or were in the clinic. And so on the left-hand side, we're looking at those compounds that have led to FDA approvals for target covalent inhibitors. The alpha beta unsaturated carbonyls, and we're demonstrating 33 examples here. 18 of the 33 did have the alpha beta unsaturated carbonyls in there. And then we can see boronic acids and esters and alpha ketoamides, the 2 other most predominant ones. And then on the right-hand side, we're looking at those compounds that are currently in development, that are currently in clinical trials. Once again, still dominated by the alpha beta unsaturated carbonyls. Then we wanted to take a look at the actual targets these compounds were acting upon. And if we look at the approved drugs, which is the donut on the left, we can see that EGFR and BTK, glucan tyrosine kinase, were those that were most popular or most predominant in those that have been approved. But you can see there's quite a distribution of other ones as well. So, 33, but there are 17 target types that are represented. And if we look over the right-hand side, this looks to be kind of an interesting, maybe change in direction that we're seeing here. EGFR and BTK dominate among approved, and they are also present in development. But now CRAS is a target that has come up and now appears to be the most dominant of targets that are currently in the clinic. Then we thought we'd take a look at therapeutic areas. So for the approved drugs, 2/3 are targeting some form of cancer. And if we look at the pie chart on the right-hand side, these are the ones that are in the clinic. That still is the case. Still, in this case, over 75% of them are studying that as the therapeutic indication. We then went down to the actual specific target level and looked at the top 20 targets in the area of target covalent inhibitors that are appearing in the literature. And you see we've even broken it down to the blue bar being the journals and the yellow bar being the patents. Here are the top 20 that we found in the literature. And we thought that we would point out the fact that the kinases that have been put in the red box can often be activated in human cancers, and they do appear among 6 of the top 9 most published here. And then the other 3, the proteasome peptidase complex, KRAS and caspase, have also been investigated as potential ways to address cancer indications. All right. So with that, I will summarize some of the takeaways that we've seen in our study of the publication trends of the target covalent inhibitors. Clearly, it's something that has been consistently on the rise over the last 20 years, and that the ability to target covalent bond has several benefits. And I think we'll hear more about this in the next talk as well. But some of the ones that came out were its efficiency, its specificity, and fewer side effects as compared to targeted covalent, compared to non-covalent inhibitors. The alpha beta unsaturated carbonyls have dominated publications so far, but there are a variety of warheads that are being employed. It's a fairly Lipinski compliant class of compounds with 82% either having zero or only one Lipinski violation. And looking at the ones that are either approved or in the clinic, 73 of them have been used as cancer therapies. And then an area perhaps that's shifting here is that EGFR and BTK were the most common targets of approved target covalent inhibitors, but KRAS has now dominated when we turn our gaze towards those things that are currently in the clinic. And so I'd like to thank everyone for their attention here and just point out that we, as was mentioned by Angela up top, do look at a lot of scientific literature and the biggest and most compelling new discoveries that are coming out. And so if you would like to see some of the research behind these that we put together, please go to cs.org/insights to take a look at these. So with that, I will turn it back over to our moderator.
Thank you so much, Gary, for giving us a very nice overview of the field involvement. I think certainly the new technologies and how we can design the covalent drugs more specifically fitting into the target protein has greatly eliminated many disadvantages of traditional covalent inhibitors, which is very promising. I know there are a lot of, we call it undruggable targets, still it's a challenging for us to find affordable small molecule inhibitors to those target proteins. This is certainly an alternative way that many researchers in the field may consider that may give us a different result. So thank you so much for your sharing, and we'll come back to you when we are in the Q&A section. So our next speaker is Dr. Neil Devon. Neil is the co-founder and CEO of Totus Medicine, where he focuses on combining small molecule design, structural biology, genetics, biochemistry, and cell biology to design new classes of drugs to target untreatable diseases. Prior to founding Totus Medicine, Neil co-founded Dual Therapeutics, which is a venture-backed biotechnology company developing the first phosphatase agonist for cancer patients. Neil co-invented and helped develop the lead drug program, small molecule agonist of a phosphatase called PP2A, which led to a collaboration deal with BMS, valued over $200 million. That's very impressive. Neil, I'm going to hand you over the microphone and you may start. Thank you so much.
Great. Well, thank you, Angela, so much for that introduction. And Angela mentioned the word undruggable. So I had to add in a slide about how I think we as an industry, especially with covalent drugs, are going to change the use of the word undruggable. And so I did what I think all of us need to do when we have to answer a question like that, and entered into ChatGPT for an answer. And I think what we find there is that I think with innovations like covalent drugs, we're going to find that we're going to change this word of undruggable into a word like intractable or high-hanging fruit type targets, because what it seems to be is that with these these technologies that, to Gary's point earlier, have been around for a long time, but now we're really able to apply these technologies at a much grander scale and able to really go after targets that were thought to be undruggable. And I wanted today to start at a pretty high level, just kind of describing where covalent drugs have come in the field. And then I'm looking forward to sharing a more detailed experience that we've had at Totus Medicines, developing the first ever highly specific covalent drug targeting PI3K alpha, one of the most mutated oncogenes in cancer, which we have now moved into clinical trials. But I think about covalent drugs and the druggable and undruggable target classes. In many ways, I think about climbing a wall in a climbing gym versus going free solo on a very hard to climb wall. We all know these targets in this space that don't have great binding sites. But what we found with these covalent drugs is that they're able to capture binding pockets and binding sites on some of the most challenging targets in the industry that have plagued the industry for decades. And really what we're focused on, and as many of you in the audience know, is that like we know these drugs have been around for decades. And Gary mentioned some of these drugs like penicillin and AZT, statins, and many of the cancer alkylating agents. And in fact, actually, when we took and looked at these molecules in more detail, specifically the modern covalent drugs that have been developed over the past decade, we saw numerous clinical and development advantages from these drugs, where the percent of approved drugs was about 20%. The time from IND to approval was 6 years versus 10 years. And a huge number of the drugs on the WHO's list of the most safe and essential medicines in the world are actually covalent drugs. And what's we found more recently is the just consistent message is that this mechanism of action, by being covalent, this can actually lead to differentiated clinical benefit for patients. So what we've seen with cracking targets like KRAS in lung cancer, what we've seen where head to head non-covalence and covalence were tested. And we found that covalent drugs led to twice the duration of action in the clinic for EGFR mutant lung cancer patients, as well as covalent drugs that target non-protein targets and intercalate and covalently target DNA or RNA. And so what we see overall in this field is an incredible opportunity to go after some of the most challenging targets, specifically in oncology at Totus Medicines, but also beyond oncology in a variety of different diseases, all due to the fact that this ability to form a covalent bond with a target leads to improved potency. It can lead to an actual binary specificity to the target of interest because you can create a drug that only targets a very specific amino acid in only one target. And so it only covalently hits that target versus a variety of others. And there are numerous PK and PD advantages of covalent drugs. And this is why, over the past 2 decades, covalent drugs have represented some of the most impactful drugs for patients. And we actually now have the data where covalent drugs can lead to a highly differentiated clinical benefit for patients. And so with that, I wanted to turn the page a little bit and share our experience with covalent drugs, where, and Gary and Angela had mentioned this, where covalent drugs, there is and still remains to be stigma in the field around these molecules. But what we're seeing is that through a more detailed understanding of how covalent drugs work, a more detailed understanding of the underlying biology of the target you're going after, covalent drugs may really represent the best in class option for a wide variety of different targets. At Totus, we have first and foremost focused on PI3K alpha. And the reason for that is that PI3K alpha is one of the most mutated oncogenes in cancer. It's highly mutated and driving a significant percentage of breast, colorectal, lung, gastric, and bladder cancers. And a major problem with this target is that when you use typical reversible inhibitors in this space that are non-covalent, what you find in the vast majority of cancer cell lines is that while you achieve initial inhibition of the target, you get reactivation of the target and the pathway. So these reversible inhibitors that bind reversibly and weakly to the target are not able to achieve full and complete inhibition of the target especially in the clinic. And so what we sought to do with this hyperactivated PI3K alpha pathway is that other than using reversible drugs, where what you end up finding is that the cancer cells readily reactivate the pathway and you get significant and continuous signaling through the pathway and therefore continuous tumor cell survival and proliferation. This is why current PI3K alpha inhibitors have not been very effective in the clinic. And clinicians and PIs and patients are really waiting for a better PI3K alpha inhibitor. What we sought to do here is target PI3K alpha covalently, target the mutant, target the wild type to completely shut down PI3K alpha signaling continuously over time to lead to far improved efficacy. And so what we found at Totus is we've been able to identify the first highly specific covalent molecule targeting PI3K alpha. And we found that unlike with reversible inhibitors, this molecule leads to significant and continuous inhibition of the pathway in a wide variety of different cancer cell lines. And this translates to far improved efficacy. Now, I want to mention here, we're seeing better efficacy in all of our preclinical studies, but one of the questions in the field, and especially around covalent drugs, has been around, yes, you can achieve better efficacy, but what is the safety profile around this molecule? Because if you covalently hit it, will cause toxicity issues. And so what's so critical in this field, because there has been, sometimes perception-based views that if you covalently hit this target, you're going to get toxicity. And those perception-based views are exactly what we saw with covalent drugs targeting BTK, which now are some of the most effective drugs for cancer patients, with covalent drugs targeting EGFR, and with covalent drugs targeting KRAS. So what we did as a company is dig deep into the basic biology around PI3K alpha, and specifically around concerns that if you target PI3K alpha covalently, you will get specifically sustained hyperglycemia that could be a dose-limiting toxicity. But in fact, when you look more deeply at the data in the field, what you find is that actually when you knock out PI3K alpha in mice, what you see is that you get actually a very mild and acute hyperglycemia effect, but you don't see chronic or sustained hyperglycemia over time. And actually, again, this was reproduced in another paper where when you have PI3K alpha present, you had a glucose level of 119. And then when PI3K alpha was deleted, you have a glucose level of 118. So no effect on glucose levels, even though the perception in the field was that if you do this, you're going to induce sustained hyperglycemia. In fact, what the field saw was that when you knock out PI3K beta, another target, which our molecule does not covalently hit, that's when you see the increase in hyperglycemia. And so why this is so incredibly important is that in follow-on papers, people took so-called reversible or non-covalent inhibitors that based on a purified protein screen are actually alpha selective and don't hit PI3K beta or other PI3Ks that potently. So the view in the field was that in a purified protein assay, if these look selective, they're going to be selective in cells. But what this paper then did is that they actually took these alpha selective inhibitors and treated cells that both had PI3K alpha present and cells that have no PI3K alpha. And what they found is that previous reversible so-called alpha selective inhibitors still inhibit insulin signaling and lead to sustained hyperglycemia, even when there is no PI3K alpha present. And so what that means is that the so-called sustained hyperglycemia induced by these previous generation reversible inhibitors are not due to PI3K alpha. It's due to the lack of selectivity of these drugs because they still induce this effect when there's no PI3K alpha present. So the reason this is so critical to the development of covalent drugs is we see this perception and stigma around covalent drugs time and time again. And it really takes a more detailed look into previous classes of reversible molecules and into the target of interest to understand the therapeutic potential of a covalent approach. And so again, what we found at the company is that previous molecules are actually not very selective. And with our covalent approach, unlike with current molecules where the window between alpha targeting and beta targeting, the off-target targeting that can lead to toxicity, is fairly small, we see a far improved window of alpha inhibition versus beta and off-target inhibition with our covalent approach. And what that's led to is that in a variety of different cancer cell lines, when we look at our molecule versus non-covalent molecules in the space, while the non-covalent molecules cannot achieve full and complete inhibition of the pathway, we actually now can achieve full, complete inhibition of the pathway and hit this target very specifically and potently in a way that has not been possible with non-covalent approaches. And what that translates to is far improved efficacy in a wide variety of actual patient-derived xenograft models where PI3K alpha is mutated, but we're seeing this across PI3K alpha mutant lung, colorectal, and breast cancers, where TOS358 in red leads to either stasis or regression, while current non-covalent molecules like alpelisib lead to progression and can't stall the tumors because they can't effectively and completely shut down PI3K alpha in a specific manner. Most notably, one thing I'd like to point out is based on our preclinical data, we're also seeing a dramatically improved safety profile. So first to note, in our preclinical studies, we're not seeing the sustained induction of hyperglycemia at doses that achieve full efficacy and targeting of PI3K alpha. And based on our 28-day GLP studies, we're seeing a dramatically better therapeutic window as compared to previous generations, specifically reversible or non-covalent PI3K inhibitors. And one thing we're very excited about now at the company is we now have advanced this program into a phase 1 clinical trial where we're actively dose escalating this molecule in a variety of patients with PI3K alpha mutant tumors, really for a major opportunity to address PI3K alpha mutant cancers across a variety of different subtypes, including breast cancer, gastric, colorectal, non-small cell lung, head and neck, and other cancers, as well as apply our molecule in combination therapies across a wide diversity of different cancer types. So with that, just in conclusion, what I'd love to share today is that this shows the power of covalency, not only its ability to lead to better target engagement, potency on target, and efficacy, but also better selectivity on target and therefore a better safety profile as well. And so with that, I'd love to turn it back over to Angela and open it up to any questions.
Thank you so much, Neil, for sharing this exciting research data. So I'm going to start the question one by one. And some of the questions may be asking to one of you. Most of the questions, I guess, is open, open-ended. And either Gary or Neil, you feel comfortable, please feel free to chime in. The first question is more about the trend. Gary, you showed a very nice trend graph, how covalent inhibitor research activity has been increased recently. And also, Neil, you showed your work actually really demonstrate, from the research end, how promised this is. So the question is about, we have had some successful covalent inhibitor drugs such as aspirin and penicillin, but since there was a long period of silence, why do you think there's a resurgence of covalent inhibitor drugs recently? What are the reason or behind, interpretation of these phenomena? Gary, do you want to go?
Yeah, I mean, just from what I've seen, I think there was just that's key moment of insight there where someone kind of sat down and took a look at the possibility of getting the combination of the selectivity from designing binding with the site of action, but then taking advantage of a electrophile that has got that fine-tuned reactivity, right? We call it the Goldilocks area, where it's not too hot and it's not too difficult to react. And then you can just look for the presence of residues in the area of the active site or the area you want to act at, and be able to place that electrophile just right in your molecule, such that it would put in the perfect position to form that covalent bond. But like I said, it's not so reactive that it's going to be reacting everywhere and resulting to a lot of side effects. But
Yeah. Yeah. And I would say that, jumping on Gary's point, when penicillin and aspirin were discovered, we didn't know they were covalent. So no one knew that these molecules were covalent. And in fact, we had known. I'm actually worried that penicillin may not have been developed in the way that it was, because at the time, there was a lot of stigma around, can you create specific covalent molecules, even though nature has been doing that for basically thousands of years with penicillin and aspirin. And so I think it took some very brave scientists to really push covalency and build out some of the proof that these could actually be effective drugs, and do that in a rational manner, as Gary pointed out, to really, in a couple of successes have now led to, like, I think, a very exciting moment for the covalency field where a lot of companies and pharmas are diving in.
Thank you. I think it may also matters with, other surrounding technologies involvement as well. For example, structural biology has made huge progress in recent years, and there huge advancement in computer-assisted drug design to review or to artificially green huge amount of fitting between chemical structures and target proteins. That may also help us to improve the efficiency. Yeah, but great points. Thank you so much for sharing. I'm, going to read the next question. I think this is for Neil. Do you think covalent inhibitors can be applied to other precision medicine targets? You mentioned in PI3 kinase, there may be, patients with specific mutations. Could these be beneficial?
Yeah, I think there's multiple precision medicine oncology targets that are ripe for covalent targeting. And I describe them in two large classes. One is targets that have developed resistant mechanisms to non-covalent drugs. That sits in the class of targets such as EGFR, but also going after some of the known cancer regulators that we have not been able to drug for decades. things like transcription factors and transcriptional regulators that we know drive a huge percentage of cancer. But reversible and non-covalent approaches just have not been able to drug these targets. And so I think we're going to see a huge amount of work done specifically on covalent drugs going after those types of targets in oncology that are known drivers, but undrugged with current technologies.
Thank you for sharing. I know there are a couple very notorious oncoprotein, but we never had any good inhibitors to them. Like MYC is, making many copies in many types of cancer. So far, I haven't seen a good MYC inhibitor. Perhaps it's due to its surface structure. So we'll be very, look forward looking for future these type of inhibitors. Yeah.
Targets like MYC, beta catenin, the
Yeah.
E2F transcription factors. We need drugs towards these targets. And I think that's really, those targets are going to represent the future targets for the oncology field to go after. And I think covalent drugs are going to play a huge part in that.
hmm Yeah. Great thoughts. Well, in the past, we have been looking a lot on the like kinases. Kinases are a bunch of proteins are relatively easy to design inhibitors. But Nick, when I was reading your bio, I'm also impressed you looked at the phosphatase as well. This is another group of protein that have been neglected due to various reasons, right? I'm not sure. I don't know. Covalent inhibitors will play a role there. Maybe not. But I'm gonna go to the next question, Gary. This is for you. One of the audience are curious about um, how the covalent inhibitors can covalently link to the amino acid. You mentioned cysteine was mentioned in one example. Are any other example amino acids that people, routinely design to link covalent linkage?
Yeah. So, I mean, obviously with something like a microacceptor, which alpha beta saturated carbonates are, there's a variety of nucleophiles that could perform the task. Perusing the literature, cysteine stood out in that, I think in well over 2/3 of the cases, if you look at the ones that have been approved, what have you, it is acting through a covalent bond form with cysteines. But we've also seen among the approved drugs, ones that are acting at serine and threonine as well. Um, so, but like I said, I don't want to say that is limiting. Let's face it, this is a fairly new field with its first marketed drug approved in 2013. So I think there's a lot of other potential out there, and hopefully, a lot of brilliant minds like Neil working on it, that I'm sure they'll be able to expand that. But if you're asking about what's come out so far, it's dominated by cysteine, but then also seen some approved drugs acting through serine or threonine as well.
Neil, would you like to add something?
Oh, completely agree with Gary. And I think that a big question and opportunity in the field is the, as Gary said, non-cysteine. So I would only reiterate what Gary already mentioned here.
Great. The next question is about expand these covalent inhibitors to other types of drugs. I think these people from audience mentioned two important disease, such as in the area of infectious disease like TB or malaria. If there's any research actively ongoing designing covalent inhibitors for TB or malaria. Neil, Gary, Neil, maybe you can go first.
Yeah, certainly. I think it's an area where covalent drugs can make an enormous impact. Most antibiotics, a lot of antibiotics, at least the first generation antibiotics, were covalent. And by covalently hitting targets, that leads to clear advantages from the, the potency, specificity, as well as just like the duration of the effect of these molecules because they stick to the target. Now, so I think what we really, I think a big area for covalent drugs, and for example, some of the COVID drugs were working through a covalent mechanism of action, is going to be in infectious disease and going after some of these proteases and other targets that we can now hit covalently. I think what's limited that to some degree is as of, a couple years ago, we really didn't have high throughput technologies to screen covalent drugs at scale. Now we've seen a new and complete renaissance in the use of proteomic technologies, in DNA encoded library technologies that Gary knows a lot about as well, to screen these types of drugs at far more enormous scale than has previously been possible. So what that means for the field is that we don't only have to go after rationally designed covalent drugs anymore. We can screen covalent drugs at a completely far more dramatic, higher scale and find these drugs through those mechanisms in a way that wasn't possible really 10 years ago. So I think that's going to be really exciting to crack into new targets beyond kinases like you described, Angela.
Thank you, Neil, for your share. Gary, would you like to say a few words here as well?
Well, in this instance, I would like to say that I thought Neil covered that pretty well. You know, uh, what we sit here in the year 2023, and we know the technology we have at our avail, and we say we can do this and we can't do that. Um, I think that this technology itself has opened a lot of eyes to target covalent inhibitors. Is a very big and promising new field, and just the science and the capabilities of all aspects of drug discovery are being refined and enhanced, and we can look at bigger data answer sets. I don't know if we've really mentioned AI and ML now. I mean, we can mine a lot of data that's out there with these algorithms, and little patterns that were kind of invisible to us previously just because of the sheer magnitude of data that's out there. Now we can tease this stuff out. So, yeah, once again, that's not a real concrete answer, but I think that it's definitely a potential. We need to take advantage of all these new technologies that are presenting themselves to push drug discovery into that next level.
Thank you, Gary. Yeah. My next question is, what are some of the most exciting new technologies to discover the covalent inhibitor drugs? Um, I can give you a few examples. New exciting news. When I hear that the KRAS inhibitor was approved, that was a big deal, exciting news to me. Because this is the oncoprotein has been around. I used to teach oncology, cancer biology at universities. So I know how bad this oncoprotein is in various types of cancer, and we show statistics number. How many patients have mutations. A lot of those mutations on the exact same spot. Now we haven't approved the drugs to this exact oncoprotein, so that's fantastic news. Another example may be the COVID. We have this drug developed in super speed, to covalently inhibit the protein from the virus. So that's fantastic news. How it really shows effectiveness and the possibilities. Yeah. But I leave the door open to you both for you to share your exciting news from based on your research, and Gary also from your work as well.
Yeah, definitely. I think the discovery of the covalent KRAS molecules from Kevon Shokat's lab really have changed the game in how people view these drugs. Now, what I would say is when those original discoveries were made, and it's been almost 10 years since they originally identified that you could covalently target KRAS in this manner, it was using technologies that work on a level that we've surpassed now, 10 years later, by orders of magnitude. Specifically, proteomic technologies have completely and exponentially increased in their sensitivity and their throughput. So now what I'm really excited about is technologies, yeah, like proteomics, like utilizing array-based mechanisms, like utilizing DNA encoded libraries to screen for these molecules at scale. where it took, actually a number of years to take those covalent KRAS inhibitors and turn them into drug-like molecules. My hope is that with these new technologies and the efficiency that they deliver, we could turn that into a year or two, or even someday, even shorter than that, where we could rapidly get to drug-like molecules, like the COVID example, that can effectively hit a variety of different challenging targets.
Yeah, if I were to talk in this area, I guess I already kind of talked it a little bit. Some of my most impressive readings recently are just what AI and ML are doing for us in being able to, you know, um, generate protein structures for almost every known, alpha fold out there is be able to give us protein structures that, obviously help someone like myself, who was a medicinal chemist, actually have physically something that you can look at and design for. And then once again, with the large data sets, being able to virtually screen in areas of chemical space that we haven't even attempted previously. And all this will then shorten the screens, because what's one of the biggest Achilles heel, I spent 25 years as a drug development scientist. Biggest Achilles heel is it takes 10, 12, 14 years, even in the case where you do get something and do get it to the clinic. It's just too long. We kind of leveled off for a long period of time in the efficiency of drug discovery, and there's still, it's not a finished technology. But if you look at the promise out there in the last 2 or 3 years, we've seen some of these companies go from target discovery and validation to putting stuff in the clinic within a couple of years now. So that to me just is amazing. If we can, check off an order of magnitude in the time it takes us to actually get things into the clinic, the benefit to patients is going to be tremendous.
Yeah, truly echo with what you just said, Gary. We do have so many questions on the queue, so I'm gonna move forward. The next question is about the blood-brain barrier. This is our brain is always at the one of the heart organ, we try to target. So the question is, any investigation has been done into the most effective covalent warheads and their permeability of the compounds going through the blood-brain barrier. Any of these warhead will, hinder the permeability issue.
Sure.
Well, I mean, if you're talking about the warhead themselves, I mean, if you
Or it
look
could
at
be
it,
both. It could be
okay,
both. Yeah,
yeah,
yeah. Think
yeah, yeah.
about, yeah.
So, obviously, always the problem was if something's too polar, it's going to be tough to get it across the blood-brain barrier. I mean, obviously, the functionality that is one of the key aspects of the success of these. I don't think that should be a problem. If you're looking at alpha beta unsaturated carbonyl, that's something that should not have a problem crossing the blood-brain barrier. I will say, in all honesty, that is an aspect of this that I probably haven't spent a whole lot of time looking at. However, in the literature I did read, I didn't hear a lot of studies coming out and saying, oh, and 2/3 of our compounds failed because of the inability to cross the blood-brain barrier. I didn't see that as an issue that was routinely brought up. I don't know, Neil, if in your case,
Yeah.
you have anything different.
Well, I think we know that a variety, especially acrylamides, molecules do get through the blood-brain barrier. And actually, the real driver of the blood-brain barrier penetrance is not the covalent warhead for these molecules. It's the kind of guidance system that Gary was describing. Largely, like that is driving the permeability in the blood-brain barrier. Now, what I do want to point out, I think covalent drugs are fantastic to target brain targets. And the reason is because the main issue with the blood-brain barrier is efflux, right? Efflux out of the brain. But what covalent drugs can do, as long as they get in, they can covalently hit the target. So even if they're efflux, once they stick to the target, they're there and having their effect. And so we've seen multiple examples, specifically, for example, Tagrisso, the covalent drug targeting EGFR, where it's led to actual responses in brain metastasis, because it can get across the blood-brain barrier, covalently link and hit the target continuously, because once it hits, it's there forever. and actually lead to benefit. So I think blood-brain barrier issues really using covalency to solve those and go after those where you don't need as much brain penetrance at the end of the day because you have a covalent drug is a great area to focus on.
Great thoughts. Thank you, Neil. Thank you, Gary. I also think, going to the brain is a drug delivery problem, right? We could also have, there are many tools available to enhance the permeability of the drug going through the BBB. Um, you can add various modifications to the molecule, make it more membrane permeable, or even lipid nanoparticles, for example, they also lipid tails, you can modify them. Um, but certainly covalent drugs, it like the guidance system, right? If usually the molecular weight is smaller, they're gonna be, more likely to be able to cross the barrier. Um, yeah. All right. So let's move on to the next question here. Um, I'm not sure if I will be able to pronounce the protein name correctly, but Neil, this is a question for you. There are a few overlaps of PS3 kinase with a protein called plasmodium falciparum. I think that this probably is a protein from malaria. Are you looking forward to evaluate your molecule against malaria, especially the PF? Or maybe this is a strain of the malaria.
Great idea. We haven't, but now I'll go look it up and we'll take a look into malaria. Because there's an opportunity to make an impact there, of course. That sounds really exciting. So thanks for the idea.
Great. The next question is about the Lipinski rule. I know, Gary, you mentioned that, and this is an open discussion for Neil, for you as well. The question is, first question, basically, what is the Lipinski rule? Second, maybe a follow-up question, maybe more meaningful for medicinal chemists to discuss, how much should we follow the Lipinski rules in these days drug discovery world? I'll open it to you. Gary, maybe you can go first.
Yeah. So the Pinsky, often referred to is the rule of five. So you're trying to uh, look at your number of hydrogen bond donors, number of hydrogen bond acceptors, your molecular weight, and keeping the log p under of five. So it's been a rule of thumb that has been around probably for 40 or 50 40 years now, anyhow, to kind of serve as a guidance. Now, it's not to say that there aren't approved drugs out there that violated one or more of those rules, but it's typically um, when you look at what's involved in the rules that Lipinski stated, it's kind of some common sense going on. So, but we have discovered ways through liposomes and what have you that if that is an issue that we can't address, the science out there in delivery has made leaps and bounds in giving us different ways that we can get around those issues and get to locuses of action. Even in designing some of that targeting into the liposomes themselves to help the material get to the appropriate area, that type of thing. So, anyway, that's kind of what my thoughts on the area are.
Yeah, and similarly, I would say the rules of five are really guidance, and I would encourage a lot of scientists to look at them as guidance and not simply reasons just to disqualify potential molecules. And one of the most illustrative examples of this is what we've seen with the bifunctional PROTAC field. Those really do not comply to the Lipinski rules in any way. And it's an excellent potential modality that can make a real change in patients' lives. And I think, based on Lipinski rule view, those molecules would never have appropriate pharmacokinetic or dynamic properties. In fact, they do. And so this issue is a little bit more complex. So what I encourage is, you know, being willing to step out of Lipinski and testing your molecules in appropriate biological assays that get to these real questions of, is it cell permeable? Is it orally bioavailable? Can it get appropriate exposure? That's what Lipinski really meant to get to, right? With his rules. But I don't think his rules are definitive, and there's just too many examples where they're not. But they're fantastic guidance. Like if you can make a molecule with your given functionality within Lipinski's rules, there's no reason to move out of that. But if you can't, absolutely, you should move out of that and make sure to like focus on testing and following the science really around those molecule properties.
Thank you so much for sharing about. The next question is about the resistance. So we know, especially in treating cancer patients, you know, human body gradually develop resistance to various, drugs over a period of time. Do you see any advantage of covalent inhibitor drugs to have less likelihood of developing resistance? Neil, maybe you can start.
Yeah. So what we've seen in the clinic is times where covalent drugs were tested head to head with non-covalent drugs in patients. We've seen that covalent drugs have a better duration of response, which means it takes longer to develop resistance. Now, to many may seem paradoxical at first because these molecules that are covalently binding bind through one amino acid, right? At least covalently bind through one amino acid, but interact with many more. And so one of the resistance mechanisms that can develop is for example, cysteine that these covalent molecules are binding to may mutate into another amino acid, rendering the covalent drug. The problem with that is that generally with non-covalent drugs, which bind weakly to a larger pocket, basically almost any amino acid in that entire pocket can get mutated to drive resistance mechanisms. So what we've seen is when you have situations where there's a lot of resistance mechanisms that can develop to your non-covalent approach, you do develop resistance a lot faster. You have more chances to develop resistance. Well, with covalent approaches where they're binding so tightly to the pocket and through that cysteine, there's fewer resistance mechanisms to develop to these covalent approaches. And therefore, we've seen from the clinical data, specifically with the head-to-head trials looking at non-covalent versus covalent EGFR inhibitors, that the covalent EGFR inhibitors lead to a better duration of response. And therefore, it takes longer to develop resistance.
Yeah. Thank you, Neil. I think maybe Gary, do you want to share? Um,
No, really, I can't say anything beyond what Neil said, and
yeah.
that's the one thing that I came away with. And I was actually thinking through this while Neil was presenting his slides. Why would that be the case? And I think he came to the same conclusion I would. And it's just that, where for a given molecule, it could be 15, 20, 30 residue mutations. Any one of them can give you an issue. Now, you're only worrying about one. So, but yeah, I mean, that was a great set of experiments and yeah, I think you really, really fully covered it. I don't have anything to add.
Mhm. With another thoughts, just from my uh, naive guess, right? The covalent inhibitors usually they require less dosage or lower concentration to be effective? Would that be a reason they're less likely for developing resistance because you're more focused, basically?
Yeah, I think it's they certainly require less of a dosage. And I think what's really driving the decrease in resistance is just at the end of the day, if we bind the target so much more tightly, it's
Yeah.
hard to develop mutations to prevent that versus binding the target
Mhm.
weak. So, but just by hitting the target so hard and so tightly, it makes it harder to develop those resistance mechanisms.
hmm Okay, great. So we do have a few more questions to go through. The next question um, is that there are a lot of fluorine in drugs. Any specific target for fluorine? How does that affect covalent bond forming?
And maybe the question is around fluorine and targeting fluorines.
Um, there, well, there are a lot of fluorine in drugs. I guess it's about how fluorine is an atom with a functional group to form any chance it may form a covalent bond.
I don't know,
Well,
Gary.
yeah, yeah,
Yeah,
that's going to depend on exactly where it resides. Um, you probably need to have a fairly activated methylene around that carbonyl to get that to be something you have to worry about seriously. Um, Obviously, you can undergo SN2 and what have you, but even in the warheads that we saw that do incorporate fluorine, they had to put an activating group next to the methylene or electron withdrawing group in order to get that be an effective way to form the covalent bond. So unless you've got a very reactive fluorine moiety within some of these drugs that do have fluorine in them, I don't know that I would predict firsthand that would be something to worry greatly about. But I don't know, Neil, your thoughts?
yeah, and I just was thinking about this while you were talking, Gary. It's like there have been some, maybe this question is related to some acrylamide, fluoroacrylamide kind of molecules that have been developed more recently to tune the reactivity of covalent warheads. So potentially that is an area where for certain targets, I think tuning the reactivity of the warhead, there's a lot of things you can do there. If you start with very reactive warheads, and adding different substituents is a key part of that. What I'll say there just more broadly is like, it's not like there's only 10 or 15 warheads you can use. Like every substituent you put on a warhead can make it completely different. So even with acrylamides, if you attach it to different things like an alkyl group or an aryl group, will have completely different reactivity. So I think there's a huge opportunity to go after, a wide variety of different constituents on warheads, and probably things like we haven't even discovered yet from that world of kind of substituting warheads with different functionality to enable tuning and increase or decrease reactivity.
Thank you for sharing. I think next question is exciting. I like that idea. It asks, do you think the target covalent inhibitors can be expanded to newer modalities, such as PROTACs, which bind to protein for degradation, induce degradation, and can continue the cycle? The advantage of not requiring a strong binder would be very advantageous here. So it not only bind, right? But also the PROTAC actually system helps with you to identify the target protein precisely.
Yes, absolutely. And I think that's a huge area of interest for the industry right now. I think, namely, Dan Nomura's work at Berkeley with Novartis has a huge focus on covalent PROTACs. And because that offers an ability to kind of take in like a second order or third order mechanism, maybe to like a more classical approach with a covalent approach. One of the things that comes to mind also is, can covalency be applied to beyond small molecules in general, to biologics, for example, to antibodies, to peptides, to RNA, to DNA? There's real potential there. And I think it's going to take some time to get there, bringing biologists and chemists together in some of those modalities to apply some of this. But at the end of the day, we're all trying to do pretty much the same thing in many ways, like create drugs that very potently hit the target we want it to hit and don't hit other things in patients. And so with these different modalities, I think covalency can and will play a role.
Thank you for sharing. Gary, do you have anything to add?
No, and
Yeah.
the only thoughts that I had that area was exactly the things you got to near the end then, and that is incorporated in therapeutics. Like we see this explosion of RNA therapeutics out there now, that this could really give you a real nice fine tuning and increase the specificity of action if you would be able to call it into use something like a nice warhead. So
Uh-huh.
yeah, otherwise I thought he covered all the
Yeah.
other possible avenues that, really well.
Yeah. Well, as biologists, we always think, the biological way is the most specific, with high specificity. So I was like, well, antibody is specific enough. Do you really need covalency? Maybe, covalency will enhance the specificity. And even nucleic acid, they usually bind pretty specific, but that certainly can eliminate off targets. There are still off targets in certain cases, like CRISPR. Off targets will introduce significant side effect, although the ratio percentage wise may be small, but still, so great thought. Yeah, that really opened my mind. Now, while you're talking,
Mm.
this question keeps coming to my mind. I think screening probably is still a quite, effective way for identifying new molecules, such as, with covalent activities. Is there or are there, active resources that people or research lab routinely use as the library collections for you can screen for various, protein targets, whether they have been conjugated with covalent warhead of a variety of small molecules, or maybe those are not, if not in the real, chemical substance, perhaps in the silicon mode. Are there any active research ongoing with preparing those type of library molecules?
Yes, definitely. I think, and we come from the view that the ability to generate significant amounts of reproducible wet lab screening data is going to mean the difference in drug discovery and drug discovery campaigns. One thing that I'm very excited about is that we have seen a kind of burst of work done by a number of CROs, et cetera, that have made available very large chemical libraries. Like Gary knows well, 2 decades ago, if you wanted to buy a Dell library, it was pretty hard. And when I say Dell, it's DNA encoded library. Now, if you want to just go out and buy a DNA encoded library to run a screen, it's pretty straightforward. And that's really exciting. So, like, almost any lab in the world now can get a hold of tens of millions to hundreds of millions to billions of molecules to run a screen. What I will say there is why we maybe haven't seen like a complete, you know, changing of the field is that the screening techniques are still very challenging. What I would hope is that someday we could, have a technology like Illumina for next generation sequencing, where you could screen billions of molecules in a cellular setting against targets very rapidly and reproducibly. That is something that Totus is working on doing, but I think that we're going to see a wide variety of different technologies develop going after exactly this, like being able to access these huge libraries that are publicly available, which I think we should all be really excited about in the field, but then being able to utilize those libraries to screen and understand what they're doing in new ways and build out these massive data sets that are high quality and highly reproducible for this space.
Great.
Yeah.
Thank you so much. Yeah, go ahead.
Oh, no, no. I was going to say, and I know I've mentioned this twice already in the course of this question and answer session, but I've just been really intrigued with the potential once we do generate enough data, have enough quality data that's been cleaned up to train these models. You know, you talk about developing the actual biological high throughput screen. How quickly right on its heels do you think the ability to do this, in silico? Um, uh, Obviously, anytime you go into a new field, you're going to need to train a new model and what have you. But I just in some of the early successes that we've seen over the last 2 years with it, um, how I was curious to know how quickly do you think the ability to do this electronically will be right on the actual ability to do it physically?
Yeah, I think that the rate limiter in many ways is actually the ability to do it almost physically in many ways. So what I'm indicating there is just like, I think once we can do it physically, we'll also be able to do things that are incredible electronically. And the reason why is that the computation aspect is dependent on the data. So what
Yeah,
we need to do is be able to generate massive amounts of high quality, reliable data that's highly kind of organized, and then that will unlock the abilities of AI and ML in this space. I think it's fair to say that there's been a lot of excitement there, but we haven't seen that translation to patients. And I think there's been like a bunch of Nature reviews published over the past 2 weeks indicating that, unfortunately. But I'm very confident, to Gary's point, that is going to change with multiple, whether it's academics, pharmas, or biotechs in this space, who are starting to grasp the importance of the data and it being highly organized and structured, and then utilizing that to discover drugs that we probably didn't think were even possible a decade ago.
Thank you, Neil. You're actually speaking of one of our core competencies at CAS. So we do, work on data, collecting those data, curating those data, preparing the data, make sure they're high quality, also standardizing them in a way that data are harmonized with each other. So that's another important aspect for preparing the, machine algorithms or, artificial intelligence type of work. Um, yeah, so CAS is one of the, active dedicator to this field. Especially in the chemistry and the chemistry related, research areas. Great thoughts. Yeah. Looks like we have time for the last question. This is a question about off target side effects. We know covalent inhibitors, as they design, they're most likely going to go to the target protein, but we can never prevent it. May also covalent may link to off target proteins. Any concerns or any ways to evaluate these types of off-target side effects? Any concerns of immune response to those off-target covalently bonded, site in our body?
Yeah, and I'm happy to jump in. Is that one of the great things about covalent drugs is you can actually do whole cell proteomics with covalent drugs. So with reversible interactions,
Mm.
there's frankly no good way to measure the reversible off targets across the proteome. You
Mhm.
really don't know all the reversible off targets off the proteome. And it's almost like a situation where because we can't measure it, we don't think about it in this industry, but it's there, right? But with covalent interactions, you can utilize proteomics to really understand and look at all of the covalent off targets. And that's been incredibly useful for the space where these molecules have been dramatically more specific than people would have expected 30, 35 years ago. That also being said, is like we're now at a day and age with covalency where we kind of know which warheads are highly specific, which warheads are highly reactive, and which warheads can turn into FDA approved drugs from a safety and specificity perspective, but also like from a manufacturing and quality perspective and shelf life perspective. We know that there's warheads that you can utilize in covalent drugs that will not lead to immune effects or major safety or any really safety issues. So I think we're in a lot better place. And I just encourage in the spaces, like just because we can measure it, we always have to remember there's also reversible drugs have off targets, but you just can't measure it. But that doesn't mean they're not there at the end
Yeah.
of it.
Great share. Yeah. Looks like we have time for our last thought, to the audience. Would you like, maybe Gary, would you like to share your last piece of, critical message you would like the audience to take back home?
Yeah, no, I just was very interesting session. A lot of great discussion. Was really excited to see the depths of Neil's work and all the promise that's come forward, not only in its efficacy but its safety. And with all your fingers crossed from as it enters in the clinic. And just if I go and think about the area of targeting covalent inhibitors overall, one little stat that I kind of forgot to mention that I thought is kind of telling is over the last 10 years, we've averaged about 43 FDA approvals a year. And so in those same 10 years, at least 33 of them have been targeted covalent inhibitors. So you're talking 7, almost 8%. And this has just started, right? I mean, the first approval was in 2013. I think a lot of eyes are being opened. I think, like Neil said, a lot more labs, a lot more companies are going to start looking into this. And I'm really excited to see where this goes. And if this becomes, one of the main tools in a drug discovery scientist tool belt, it is the use of targeted covalent inhibitors. So,
Thank you,
Yeah,
Gary. Neil? Yeah.
Gary, very hard to follow up. But I think as you mentioned, it's a really exciting time for covalent drugs in this space. And I just encourage people to, jump in and dive into this space. There's so much more to do here. And I think if you're interested in drug discovery and development campaigns, especially across challenging targets, covalency is just a fantastic place to be, technology to utilize and develop. And so I think we all hope that there will be more covalent drugs coming to the clinic, getting approved over the next decade, and have a lot of conviction around that.
Thank you so much. Well, my take home message would I would like to encourage every one of us here come back to visit us often. ACS webinars. Here you will find a lot more interesting topics that you may never see in your daily radar. Right. Also come to visit us at CAS Insights, where you will find a lot of emerging scientific topics being discussed and also presented in a landscape view, which can really help you to broaden your vision and scope. So I hope today's webinar is helpful to your research, and we'll see you next time. Thank you so much, Neil and Gary, for joining.
Thank you for watching this presentation. ACS Webinars is provided as a service by the American Chemical Society as your professional source for live weekly discussions and presentations that connect you with subject matter experts and global thought leaders concerning today's relevant professional issues in the chemical sciences, management, and business.